Well, we're going to get started with our first set of lectures on how a neuron actually works. And as you can imagine, it's a pretty complex process. What a neuron does, not that complex, right? What a neuron does is just send information from one part of the body to another. But that's not that easily done when you're in the neuron itself. So we got to talk about what's happening inside and outside the neuron as it's being used. And I'm basically going to break it up into a few different stages. And the first thing we're going to talk about is something called resting potential. We're going to talk about what's happening in the neuron when it's not being used. And surprisingly enough, there's a lot happening when it's not being used. So that is called resting potential. So let's get started. So basically, a resting neuron is one that's not sending an impulse. So there's very few neurons that are not being used at all. They're usually being switched on and off constantly. But in the case of what we're going to talk about today, we're going to say what's happening when the neuron is not being used. And this entire process is really about just one thing: controlling ion movement. It's all about moving ions from one side of a membrane to another and having that happen at the right time in order to cause this electrical potential to occur. So we're going to use some terms throughout this unit. The first one being polarized. So what we say is when a neuron is not being used, when it's at rest, when it's resting potential, it is going to be polarized. And the reason why is because there's an unequal distribution of ions on either side of the membrane. What? Let's get into this a little bit. So you're going to be using this diagram a lot in this class, in this section. I like this diagram. It's not the most beautiful thing you've ever seen, but it does the job. What are we looking at here? We're looking at a neuron, right? I'm going to be referring to intracellular and extracellular. Extracellular means it's outside of the cell. Intracellular means it's inside of the cell. This is happening, and action potential happens in the axon. So everything that I'm going to be talking about today is going to deal with the axon. And let's take a look at what's happening here. There's a handful of things going on inside this axon. First of all, we have these yellow circles, and these little yellow dots represent a sodium ion. And the sodium has a charge of a +1, right? Our red little dots here represent potassium ions. They also have a charge of +1. So if they're both positive, how could one side be negative? Well, we're going to be using the term potential in here a lot. And we're going to be using the word polarization, depolarization, repolarization. What this refers to is the difference in charge from inside and outside the membrane. So just because one of these is not negative, you can still have one side that's more negative than the other. If I have more positives on one side and less positives on the other, essentially one side is negative and one side is positive. So the first thing I want to make sure we all understand here is when I'm talking about charges on the membrane, the charge that I'm talking about is the relationship between the inside and the outside of this membrane. So what we see here is we have our membrane. There's also some channels in here. We have these blue boxes that represents potassium channels. We have these little green boxes, they represent sodium channels, and we have this gray blob here, which is the sodium potassium pump, which I'm going to be talking about a little bit today. But at this point, one of the things I want you to understand is this idea of polarization. When I say the word poles, hopefully you think of something like this, right? If I have the Earth here, if I have the North Pole and I have the South Pole, those are as far apart from each other as they could ever get. The more far apart they are, the more polar they are. And that's what we're talking about here when we talk about the relationship between the inside and the outside of this membrane. This is going to be called polarized because the inside of the membrane is going to be more negative compared to the outside. And there's actually going to be a number attached to that, which we're going to be talking about today. The resting potential is going to be negative 70 nanometers. So if I were to hook this up to a graphing device, if I were to try to get like we did with a myogram, we talked about that before, you can do the same thing with a neuron, and you can graph it out. We find that this is negative 70 millivolts compared to the outside. So the outside would be 0, and the inside would be negative 70. That is very polar because they're far apart from each other. So if I have 0 here, and I have negative 70 here, this would be polar. If I make it negative 80, this would be more polar, right? That would make it more polar. And if I make it negative 50, that would make it less polar. So I just want you to understand the idea that the farther apart these charges are from each other, the more polar they are. So let's go back to the notes here and work through this. So basically we say that this is going to be a polarized membrane because there's an unequal distribution of ions. There's going to be more positives on one side, less positives on the other. So let's get more specific here. Outside the neuron, we're going to have a greater concentration of sodium ions. And inside the neuron, we're going to have a greater concentration of potassium ions. And we're also going to have these large, bulky proteins that are negatively charged. So let me go back to this diagram again and use this as an example. So if I take a look here, what I'll see is I can see that I have lots of sodium on the outside, and sodium has a positive charge. I have potassium on the inside. Potassium also has a positive charge, but there are more sodiums outside than there are potassiums inside. These little stars, what do these stars represent? What those stars represent are proteins. And here's the thing about proteins. Proteins are slightly negative in charge. They have little ends that are going to be like OH negative that are going to give them a negative charge. And here's the problem with proteins. They're too big to get out, so they're stuck in here. So what you end up with is a scenario where you have more sodium on the outside, so there's a whole bunch of positives on the outside, so there's going to be positive charges on the outside. There are potassiums on the inside. Now, here's the thing. Remember, when we talk about diffusion, sodium wants to get in, but sodium can't get in because the doors are closed, so the sodium stays to the outside. The potassium wants to get out, and the potassium can't get out, so it has to stay on the inside. And then finally, these large proteins that are slightly negatively charged, they also want to get out, but they can't because they're too big. So we end up with a scenario where you have sodium on the outside, potassium on the inside, and they're both stuck where they are. Let's keep talking about this a little bit. Okay. Now, one thing I just lied to you about, though, is the idea that potassium can't get out. There's different kinds of channels in which these ions can pass through. And these ions can go from one side of the membrane to another through these protein channels. And now this should hopefully be a bit of a review from biology class, but these protein channels are what lets things in and out. So there actually are some small leakage channels. Now, I hate that term, leakage channel. It just sounds kind of gross. But there are potassium channels that stay open. And those potassium channels are called potassium leakage channels. And what that does is it lets some of that potassium get out. So if I go back to my diagram here, this potassium wants to get out, and it is able to get out. It is able to leave. And when it leaves, what it takes with it is it takes its positive charge. So there's really three factors involved in the charge on the membrane when it is at rest. One is that sodium is outside and it can't get in, so it's more positive on the outside. Potassium is on the inside, and some potassium can get out. And when it leaves, it takes its positive charge with it, making the inside more negative. And then finally, we have these large, bulky proteins that are also slightly negative, and they cannot get out either. All those factors together are going to cause a scenario where you have a net charge where the inside of the membrane is negative. And specifically, it's around negative 70 millivolts. Now, that number might be different in different textbooks because it's not a set number. It's an average. Sometimes you might see it as negative 60, but negative 70 is how I teach it because that's how the textbook teaches it. But the term for this is resting potential. And if you look at the diagram down here, it shows you that. We have our sodium ions outside, can't get in, making it more positive. We have our potassium ions inside, some can get out, and then we have these large protein molecules, which gives us an overall charge of around negative 70 inside versus outside. Let's just keep going here a little bit just to make sure we understand what's going on here. Okay, how do these ions actually move? Well, ions are able to go from an area of high concentration to an area of low concentration. That is the law of diffusion, and that is really the mechanism that makes this whole process go. If there's more ions outside, they're going to want to get in. If there's more ions inside, they're going to want to get out. And the way we control that is by having these channels. And these channels are going to be either open or closed at different points of this process, and that allows these sodium and potassium ions to go in and out. So the ions enter and leave through these channels or gates that are specific for each ion. So there's sodium channels and there's potassium channels. Sodium can only go in and out of the sodium channels. Potassium can only go in and out of the potassium channels. Now, there are really three major types of channels, which I'm just going to quickly explain now, and then we're going to finish up for the lecture. Okay, so the three types of channels are passive channels. And passive channels are just an open door. They always stay open. And that is going to be like the potassium leakage channels. So those passive channels, they just stay open, and that allows some potassium to get out, which is what makes the inside of that membrane slightly negative. And then the other two types of channels are either going to be called ligand gated or voltage gated. Ligand gated is opened by an actual molecule. So for example, a neurotransmitter is going to open a ligand gated channel. So when we learned about acetylcholine, there was little membrane proteins on the outside of the muscle cell. The acetylcholine attaches to that membrane channel and it opens it up. So that would be a ligand gated channel. The next type are called voltage gated, and these are going to open when there's a change in charge. So when the membrane charge changes, that's going to open up these channels. So if it's usually positive on the outside and negative on the inside, that will be closed. But if I were to flip that and make the outside negative, that would open up the channel. So there's two different types of channels when we talk about the neuron. There's going to be voltage gated and ligand gated, and each one has important jobs that we're going to talk about as we go through. But for right now, you just need to be familiar with these terms. So let's finish up here. So basically what you end up with is a scenario where the inside is negative and the outside is positive. The reason why is because there's more sodium outside, plus some of those potassium are able to leak out of those leakage channels to get to the outside. The inside stays slightly negative because those leaving potassiums make it negative, plus those large proteins that can't get out also make it negative. And we're going to finish up here with this last idea, and then we'll be done, and that is the sodium-potassium pump. Now, the sodium-potassium pump exists along all of the membranes in your cells, and they have a really important job. For one thing, it is an active transport mechanism, so it requires ATP. But this specific molecule, this sodium-potassium pump, takes 3 sodium ions and removes them from the cell and puts 2 potassiums back in. So every time ATP gets used, 1 ATP, 3 sodiums get kicked out and 2 potassiums go back in. And again, what does this do? This keeps an imbalance of charge. If we just left this whole system go, what would end up happening is it would eventually hit equilibrium. And if equilibrium gets hit, we're done for. Because if the sodiums are equal on both sides of the membrane and the potassiums are equal on both sides of the membrane, nothing's going to move. And if nothing moves, no charge changes, and essentially you die, right? So the way that we keep the imbalance of charge there is the sodium-potassium pump. And this number is very important. If it was 2 and 2, if it was 2 sodium for every 2 potassium, it would totally defeat the purpose. It would be like me saying, hey, I'm going to give you $2, you give me $2 back. Nothing got accomplished there, right? But what the sodium-potassium pump does is it removes 3 sodium for every 2 potassium. So what that does is it keeps more potassium leaving than sodium, it keeps more sodium leaving to the outside than potassium coming back in. So just to kind of reiterate that with my little diagram here, just to make sure we all understand what's going on, we have our sodium-potassium pump that is taking, for every sodium gets pumped out, it pumps 3 sodiums out and lets 2 potassiums back in. And what that does is it keeps this imbalance of charge. So we have a positive outside and a negative inside, and specifically negative 70 nanometers, or I'm sorry, negative 70 millivolts or negative 70 nanometers are what we're going to be talking about here. Okay, thank you very much.